All right, welcome back. We're going to continue our discussion here of metabolism, moving now into the idea of oxidative phosphorylation. So our learning objective here is to compare the mechanisms of ATP formation during glycolysis and oxidative phosphorylation. So, looking at the entire pathway here, we can see the central glycolysis, we feed it into a citric acid cycle, and then now there's this magical thing down here called electron transport and oxidative phosphorylation. And we're going to take a deeper dive here into what is happening. And specifically, what we need to realize is that what we are doing is essentially stripping electrons at various stages throughout the cycle and dumping them into this final pathway. Now, there are ATPs that are generated during glycolysis and during citric acid cycle, but by and far, most of it is made down here in the oxidative phosphorylation section. And something to consider is that we are not simply dumping electrons into a spot. We are dumping electrons into a chain of events that are passing the electrons from one thing to another, to another, and to another. And this is doing a certain amount of work, which we harvest as ATP. And we'll get to how that occurs a little later on. Ultimately, though, we are putting those electrons somewhere, and that somewhere is onto oxygen, which converts oxygen into water. So, a couple of terms to keep in mind is we have something called electron donors. This is the source of electrons. And this is coming from some type of high energy electron source. In our case, we're going to be talking about food, but it isn't restricted to that. For us, it is, but for other organisms, they don't have that restriction. They have other ways of getting high electron or high energy electron donors. So again, we harvest this from breakdown of biological macromolecules. So from fats, from sugars, from proteins, we glean those electrons. When all that extra energy from the electrons are used, the lower energy electrons that are left over are then dumped onto another molecule. And we generally call this a terminal electron acceptor. For us, it is oxygen. But if we look at the wide world of species, that isn't always the case, again. And finally, the energy that is gleaned from this is used by something called the electron transport chain. And what it uses the energy for is to pump protons across a membrane. And this generates a biological battery. So to put this into more of a graphical case, or a graphical representation, here we have electron donor that is providing electrons to this thing called the electron transport system or chain. This is inherently in a membrane, and you will see why shortly. And then we are putting those electrons onto a terminal electron acceptor. Now, for us, we glean those electrons from food. We turn those carbon-rich compounds into carbon dioxide and also glean high-energy electrons from those. Again, for us, we dump those electrons once they are low energy onto oxygen, and that produces water. Now, in the actual electron transport system, we have this membrane, and on one side of the membrane there are hydrogen ions. As the electrons pass through this electron transport chain, it transported those protons to the other side of the membrane. Okay? This osmotic force is something that we refer to as the proton motive force, because we now have a high concentration of protons on one side of a membrane that want to pass through to the other side. But the fact that it's occurring in a membrane means that there's only one way for them to get back through, and that's through some type of channel. We can harvest that inherent flow of electrons to do work, and that work can be of a chemical nature, such as making stuff like ATP. We can use that work to do things like transport or get stuff inside of the cell that was outside the cell. And we can also do it to do mechanical stuff. In the case of bacteria, they can utilize this proton motive force to swim. Okay? Now, we always put our electrons onto oxygen, but that's not true for all species. Some species put it onto something else, like hydrogen, or onto a sulfur to make hydrogen sulfate. Other organisms don't actually need food to get these high energy electrons. Some can essentially take water, split the water to get a low energy electron, combine that with light energy to make their own high energy electrons. And then they can further use carbon dioxide and high energy electrons to essentially reverse this paradigm and produce their own food. This is, of course, photosynthesis and what is happening in plants. Out there in the microbial world, we even have further possibilities where there are some bacteria that can actually strip electrons off of iron and utilize those. So there's actually a lot of diversity out there on how the system works, but the general components are the same. There is an electron donor, there's some type of chain of oxidizable and reducible components in a membrane that pumps protons. And ultimately, those electrons get put onto a terminal electron acceptor. Now, in us, this is all happening within the mitochondria. And if you recall, I told you that the origin of mitochondria was in fact bacteria. And so these are little mini bacteria living within us that do this job for us. Mitochondria has two layers of membrane. So there's an inner membrane that's highly folded, and there's an outer membrane. Inside of, or in between these two membranes, we have something called the inner membrane space. And inside of the inner membrane, we have a region known as the matrix. Now, when we look at the electron transport chain, there are various components, and we refer to them as various complexes because none of these are single proteins. They're actually large complexes of several proteins working together. So here we have complex 1, complex 3, complex 4, and ultimately complex 5. To put you in perspective of where we are located, here's the outer membrane of the mitochondria. Here's the inner membrane space. Here's the inner membrane. And here is the inside of the matrix. So when we dump electrons off onto complex 1, what it does is transport protons from the matrix into the inner membrane space. Those electrons have lost a little bit of energy in this process. We then dump those electrons from complex 1 onto a molecule called Q. It is hydrophobic and restricted to this inner membrane space. It can then float freely and move from complex 1 to complex 3, where it'll deposit its electrons. It'll travel through here, causing more protons to be pumped into this inner membrane space. Those slightly lower energy electrons are then dumped onto something called cytochrome C, which transports those electrons in this inner membrane space from complex 3 to complex 4. There it dumps off the electrons, which ultimately pumps a couple more protons into its inner membrane space. And the final resting place for those low energy electrons is to combine with molecular oxygen and hydrogen ions that are still present, and turn that into water. Ultimately, what this does is it builds up a lot of protons in this inner membrane space. They want to pass back into the matrix. There's only one place for them to do that, and that is through this complex 5. This complex 5 essentially acts like a water wheel, except for instead of water turning a wheel, we have protons turning a wheel, a literal wheel. And the process of turning this wheel essentially causes several proteins to change conformation, and that takes ADP and inorganic phosphate and turns it into ATP. Now, the first complex that we have here, just to give you a little bit more detail about how this works, is here are the various different proteins. Each protein is represented in a different color. So you can see a protein here, protein here, protein here, 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 another one in here, another one in here, and over here. So it's a big complex. These electrons get dumped from NADH onto FMN, right? So this is analogous to a FAD. Remember, we had that conversation here. This is all happening on that flavin ring structure, and it can pick up electrons one at a time and dump them off one at a time. This is important because it can take both electrons from NAD, but then it can pass it off onto various iron-sulfur clusters, and these are little clusters of iron atoms and sulfur atoms that form a cubical matrix. Those electrons are dumped onto the iron molecule, but the iron molecule can only take them one at a time. So it will go from here to here, and so this can keep one, give one away, and then passes it to the next one, and to the next one, and to the next one, ultimately until it passes down here where they are put onto a molecule called Q. All this electron movement, these various iron-sulfur clusters becoming oxidized and reduced, causes a conformational shift, which essentially takes an open binding site on this face and then transports those protons to the other face of the membrane. Okay, here this is referring to cytoplasm and periplasm, which is representation of how we label it in bacteria. In mitochondria, we would refer to the cytoplasm as the matrix and the periplasm as the inner membrane space.